As we learned a little bit earlier today, there is a blurring of lines today, and certainly we just heard it from Steve Wozniak as well, between the worlds of technology and ultimately biology, the human brain, the artificial intelligence. And while singularity may not be here for decades, in medicine already technology is providing a map or at least an opening for what's called targeted treatment, also known as individualized therapy. As a cancer survivor myself, I know the resonance that holds for me. And Dr. GarthPowis, who is director of the Center for Targeted Therapy at MD Anderson Cancer Center, is joining us now to explain what it means for you, for your loved ones, and our shared future. Dr. Powis?
Well, I was almost run over by a Segway out there. I don't know whether they have insurance on those things, but I'm going to talk about personalized cancer medicine, and that's the right drug at the right dose to the right patient at the right time. But before I start, I'd like to tell folks who aren't from Houston, and maybe even people from Houston, a little bit about the MD Anderson Cancer Center. It's the largest cancer center in the world. I would posit it's the best cancer center in the world. It certainly rates every year top or second with Memorial Sloan Kettering in New York. But it's located just 12 miles from here at the Texas Medical Center. And it itself is the largest medical complex center in the world. There's many institutions there. Anderson is the largest of those institutions. So the medical center has 75,000 people. MD Anderson has 17,000 people. It's actually part of the University of Texas system, but it doesn't function like a normal university. There are no medical students. It does education, but for fellows and some graduate students. But it's a $2.25 billion a year operation. A lot of patients. It's run more like a business. Just so you know about that. So, the beginning of the last century, Sir William Osler, who's a physician, made this statement here: if it were not for the great variability between individuals, medicine might as well be a science and not an art. In fact, he was very, very well known for his statements, and he's very correct on many of them. In this case, he was wrong. In fact, we know now the great variability makes medicine a science. It is not an art anymore. It is really a science, and we're understanding what that variability is. So, we all have been touched by cancer. We know people who've died from cancer, relatives, friends, and there's a chance 1 in 3 of us may die from cancer as things stand at the moment. But I want to leave you with a feeling, I think, of hope, of a real promise for the future, and strange to me, it may seem with cancer, a sense of excitement for what the future holds in our ability to deal with this disease, which was really, it's life going wrong. And so we are able now, for the first time, to control what is happening inside our cells, as you'll see. So a decade ago, you've already heard that the Genome Project first sequenced all the genes in our human genome. There are 20 to 25,000 human genes. It was more than $1 billion operation. The total amount of money, because it was both commercial, this is Francis Collins and Craig Ventnor from Human Genome Sciences. It was a mixture, but it's certainly more than $1 billion. Now, you, if you want to, can get your genome sequenced. There's a company set up in Boston, if you want to read through this, but the bottom line is, for $360,000, you can have done to your DNA what was done for over $1 billion to actually, it was a mixture of DNAs. What you do with that information, I'm not quite sure. There are certain things we can tell you, but it's information overload. But if you want to, it's there. More useful, I think, at the present time, is that you can actually learn your risk, if you like, for certain cancers or certain other diseases through relatively simple genetic test screening, which actually looks at focused disease-producing genes. And that's the sort of state of the art at the moment. But knowing all the genome and what's going on there, it gives us a tremendous insight as to how we can actually improve cancer therapy, diagnosis, early prevention, early diagnosis, early prevention. And that's what I'm going to talk about now. And I would point out to you that there's actually been a move as a bill which was introduced by Senator Barack Obama, as you see here, on Genomics and Personalized Medicine Act. This bill was introduced. It's still in committee, and I'm sure at the present time he's got other things to think about. But it is his foray into healthcare, if you like. It's to improve the access of patients and populations in the U.S. to personalized medicine, and the federal government is supposed to help in various different ways. So there's a high profile now both in Congress and in the medical institutions around the country towards personalized medicine. So how does this work in cancer? It's different to every other disease. So you imagine if you have a heart disease, you might give a different drug based on the heart receptor that you have, or a different form of that receptor. You might give a drug a different dose based on the fact that you actually metabolize it or excrete it faster. But layer on top of that the tumor, which has its own genome. It has its own completely, not completely, but largely changed set of DNA to the host ourselves. And so you have to look at both the host genome and the tumor genome. So we know now there are about 350 maybe cancer-causing genes, and we do have the ability to molecularly target our drugs, to direct our drugs against certain of these targets. We don't have 350 drugs yet, we probably have 20. We're working our way to having a drug for every major target. So, why haven't we been personalizing cancer medicine? The first thing is, until a decade ago, mostly tumor-specific targets were unknown. We really didn't know what drove the cancer. Most of the cancer drugs we had come through actually mouse screening, and they actually inhibit the general replication machinery of the cell. So there's one drug which is used quite widely, was actually synthesized by the Allied Chemical Company. It's actually a ballpoint pen ink. It's blue. And so it was found that the mice who received this, they had tumors and they lived longer, so it's an anti-cancer drug. They turned blue, but it was an anti-cancer drug. So they engineered the blueness out a little bit. Patients who get the drug, though, it's used for prostate cancer and some other diseases. They still turn blue. You know who's getting the drug. So that's where the drugs used to come from. Now we're actually focusing them on targets. But the consequences of that, and the drugs are still in use today because it takes 10 years or more to work a drug through the system. So we're still using the drugs that were developed in the older days, is that cancer therapy is toxic because it'll kill all rapidly dividing cells. That's why you inhibit the bone marrow. That's why your hair falls out. It's not very selective. Patients receive the same drug at the same dose, and then if they get sick, you cut the dose back or you adapt afterwards. That's not a good way to give people drugs. You should know what's going on beforehand. But we didn't have the information to base those decisions on. And so overall therapy has been toxic and limited efficacy. There have been great advances, but it still was not what we would look for. Why we can personalize medicine now is that we know many of the basic physiological and biological processes driving cancer. We just know what the genes are. I mentioned that the 350 mutated genes that we know about, and there are other genes that determine how fast a cancer grows that we could also target. New approaches to drug development allow us to develop drugs to inhibit specific targets. So if we know what the protein is, we can get a crystal structure, we can discover the structure of that protein and make a drug fit into the catalytic site or the target site in that protein. We can design drugs on computers. You can now screen over a weekend 50 million compounds in silico against a target. So we have much different ways of coming up with our new drugs. Examples that are already in the clinic. I don't want to leave you with the impression that we're not personalizing medicine, but we have a few examples. 6-mercaptopurine. That's used to treat leukemias, particularly in children. We know we have to adjust the dose for metabolism. Some children metabolize it faster, and others don't, and the slow metabolizers get much more toxicity if we don't cut back the dose. Gleevec. That's the first silver bullet drug. It's active against a chromosome translation where a chromosome has been broken and two proteins that wouldn't normally be together are fused together, and it's constitutively active. It's the BCR-ABL tyrosine kinase. We have an inhibitor for that, and the response rates to a previously almost untreatable disease, CML, were dramatic. Problem is resistance develops, but we know why. So we're well on the way to controlling that disease. And then we also found it's active in other tumors, GIST, gastrointestinal tumor. Herceptin, this is an antibody against the HER2 receptor in breast cancer. So many breast cancer patients will be screened to see if they have this receptor. 15 to 13% of women with breast cancer do have it, and they receive Herceptin. And 2/3 of those patients will respond to the drug. That's a great example of personalized medicine. We are tailoring the drug to the patient. Now, just to take the breast cancer example a little further, on the left-hand side there you see this is called a genome array. Basically what you do, an expression array. You're looking at the expression of every gene in a cancer sample, and then you express it in terms of colors. Red is high, green is low, and black is the difference between normal tissue. And you can see there's clearly two different patterns which are highlighted there. It either comes from the basal cell or it comes from the luminal cell. So there are two types of breast cancer. Luminal is usually hormone dependent. Basal is usually not hormone dependent. But the HER2 occurs in both types. That's the staining that you see on the right. And so that's the best example I can give you of current personalized medicine. It's successful. There are a third of patients who don't respond, but 2/3 do. It's a great example. So what does it look like in terms of how we would like to see it work? Well, the example here is we have to get a biopsy or something that tells us what's in the tumor. So at the moment you might think with breast cancer it's easy to get a biopsy. With a metastasis to the liver or the brain it's a lot harder. You can get one of these gene arrays. You can find out which genes are expressed and which are not. You can build pathways to explain what's happening. You can do non-invasive tests, imaging, which will tell you what's inside the tumor. You put all this together and then you say that the tumor has targets maybe A out of A, B, and C, and D. So you've got 4 targets you're interested in this clinical trial. You know the patient's got A, so you give them drug A. If they've got target B, you give them B, and so on. And then you treat, and then you look to see what happens. So you can see here, and then a CAT scan here, you can see that treatments A, B, and C are successful. The tumor is going away after about six months. So you keep the patient on that treatment. If you look at treatment D, it's really having no effect in that patient. So you put that patient onto the other treatment. It may be that we can't be completely sure that patients are responding because they have targets A, B, and C. So you give that patient the chance to get a response, if there is one, on the other arm. So that's the sort of thing that we would like to do. So what are the barriers? And there are some barriers. So they fall into 3 categories. It's what we won't do, it's what we don't know, and it's what we can't do. So let me just go through those quickly. What we don't do, commercial barriers. Surprising there would be commercial barriers, but you imagine a drug company is giving a lung cancer drug to every patient with lung cancer. That's their goal. You identify a subset of, say, 10% who actually are going to respond and have the target for that receptor. You've cut your market share by 90%. So, in truth, companies are driven by, at the moment, US pharmaceutical companies, they're often driven by the blockbuster drug. It's got to be more than $1 billion. So if you're going to cut that market share, there's a lot of resistance to doing personalized medicine. An often stated alternative is we're going to find the other drug that makes our drug work better in everybody. It's one way, but it's not personalized medicine. There are too many Me Too drugs. Drug companies have lost their innovativeness. Big Pharma has. They follow other companies. There are 6 or more EGF receptor kinase inhibitors. Who needs more? They're not that much different, but drug companies know that if they've got a drug, it's approved, they can go out and market it and carve out a market share. So we need more different types of drugs. There are too many underserved cancers. If it's not $1 billion market, drug companies aren't initially interested. If you know somebody with pancreatic cancer, it's a terrible disease. Survival is a year at most for most patients. It's an underserved cancer. Drug companies don't develop drugs against it. Patients don't live long enough. There's not enough patients with pancreatic cancer to make it worth their while. They may test their drug eventually in it, but they don't look at it as a primary target for their drug development efforts. And it's difficult to study drug combinations. If you've got experimental drugs, one drug company doesn't want to work with another drug company because who knows if you may become tired in some way or your drug may be shown to be worse than their drug. A lot of barriers, and you can understand it because drug companies are there to make money, not to cure patients, really. They have stockholders, they have to follow the market. What we don't know. Now, this is a map of the wiring of the signaling pathways, how proteins and enzymes and interact within the cell. It's like the New York subway. It's like it's a wiring diagram. It's very complex. But we know that if we treat one of these targets, this is a drug rapamycin, which has been used to treat a variety of diseases. It's now approved in certain tumors. We started to try and inhibit this particular target because we knew that target is driving the cancer. And the idea is that you just block that target and the tumor will shrink. What happened, however, was that there's another loop which we we sort of knew about it was there, but everybody just really didn't want to think about it, which actually goes backwards. It goes up to the cell surface, that's at the top, and then it comes down again and bypasses that inhibition. So you've actually activated another pathway, and that's this staining that's there. That's the staining of that other target which is in the way there. You can see that you've actually got an activation. And so patients did worse on this drug than they would have done without getting the drug. And we sort of knew it, but we just hadn't put this pathway together in its entirety. This approach is called systems biology. It's taken from engineering. It's trying to apply those principles of how you figure out how everything relates to everything else in a way that we can control and we can exploit. So there are things we don't know, but we can do things about it. The last thing I want to talk about is what we can't do, and that is controlling genomic instability. So on the left-hand side we have 22 sets of chromosomes. 46 in total. They're stained here in different colors. We're able to do that to stain each chromosome to a different color. But you can see on the right-hand side are the chromosomes of a pancreatic cancer in this case. And you can see by the little arrows that there's often a chromosome is broken and then fused again with another chromosome. So you've got two colors. And it's at that junction between the two that you get protein expression going haywire, because something that's controlling the expression of a gene may be driving the wrong gene. So this happens in nearly all cancers to some degree, and it's called instability. We've lost our ability to control the structure of our genome. It just reorganizes itself, and it's the cause often of resistance and tumor development once you've got that initiating event. And I just want to show you the consequences of this. So this is some work by Dr. Louis Chodesh from Philadelphia. This is actually a study that's been done in mice. It's an experimental study because that's the only place we can really do it at the moment in this detail. So what you're looking at is on the left-hand side is that we're looking at tumor size in a mouse and a group of mice, and then we grow the tumor. The red line shows the tumor's getting bigger with time, and everything is much more condensed because it's a mouse, and so everything happens a lot faster. In humans, tumor growth is much slower than this. But what happens is you let the tumor grow and then you treat it with drug A. So you turn off that gene that we know is driving, in this case it's a breast cancer, we turn it off. And half of the tumors actually go away. But half of them just stay stationary, and then eventually they take off again. So they haven't responded. They're doing what a lot of human cancers do. They stay stable. Oh, you've got a response, a partial response, and then they start to grow again, despite the fact that the drug can still be given. And this is because another tumor gene has taken over. So it was already in that tumor probably because of the genomic instability. There's lots of different combinations, and what happens is that you select out the cells that have that resistance factor, and eventually they take over from the cells that are being killed by the first drug. So that's one thing that happens. We can control that because we know what those second mutations or genes are likely to be, and so we can give drug combinations. The other thing that happens, which is more worrying, is that you go into what looks like complete remission. Well, it is complete remission. You don't see the tumor for a long time. And then eventually those tumors that responded, they come back. There's been something else that happens. That's a new mutation that has occurred in the genome, and it's now driving the tumor. And the problem you can imagine is that we don't know what that mutation is. It could be lots of different things. So what does this tell us? We have to change our ways, we have to be smarter, and we have to may have to live with cancer. We will change our ways, we are smarter, and I don't think we'll have to live with cancer in the future. I think the first step will be getting cancer to a disease like diabetes, where you live with it. But I firmly believe we'll be able to cure it through a combination of early diagnosis, prevention, and therapy when we need it. Okay, I'm going to finish off. This is a little vignette. I have an interest in quackery and patent medicines. And this is to illustrate the importance of clinical trials. So this is a print from the 17th century, and it shows somebody with syphilis. Syphilis was a scourge of the 16th, 17th, and 18th century. It was more virulent than it is now. Many people got it, and if they got it, they took mercury. Mercury was more widely used than aspirin is today, because if you got syphilis, you either got treated by your doctor with mercury, or a remedy, or you took a secret remedy. You didn't know what was in it, but usually it had mercury. Mercury poisoning was so rife that physicians couldn't tell the difference between the symptoms of syphilis and of mercury poisoning. Now, this gentleman here, it says the Spaniard was afflicted with a Neapolitan disease. So every nation blamed every other nation for syphilis. So what he's doing is sitting in a powdering tub. So the idea was that he's in this tub. It's being heated with cinnabar, mercury ore, which is underneath. So you heat that, the mercury comes up and condenses on the skin. And to make it stick, you actually rubbed yourself, or it was rubbed with lard, so it would stick to the body. So this is the origin of the nursery rhyme, rub-a-dub-dub, 3 men in a tub. How do you think they got there? It's interesting, medical students today have got no idea what I'm talking about, but there's obviously people who have used that. That must be the internet. Anyway, so that it was widely used, and and everybody believed it worked. Until the Crimean War, they did the first clinical trial, if you like. They had a group of troops with syphilis and another group with syphilis, a battalion somewhere else. One got mercury, one didn't get mercury. They got something else. No difference. Mercury is useless against syphilis, but it was widely used. So the important point to remember is controlled clinical trials are absolutely important for developing new agents, and that's where the rubber hits the road. Sometimes the best thing you can do is say, I tried and it just didn't work. So with that, I'll finish, and I'll thank you for your attention.
Dr. Powis is a relative newcomer to MD Anderson Cancer Center, but he was actively recruited and fortunately agreed to come to our medical center here in Texas. So we're very grateful that you did, Doctor, and thank you.